laboratory. And in insects, basically innate immunity is there, acquired is not there. In innate immunity also cellular and humoral immunity is there. This one, he has worked on humoral component of the insect immunity, and he has taken certain gram-negative and gram-positive bacteria, challenged the insects, and then he has seen what is happening, and those results he will present here. And this is a work picking up in India also now, and one laboratory in CCMB. Then in live insects we have done. Yesterday also we have been thoroughly scrutinized by CDMB, and they also felt very happy that this type of work is going on here. And I suggest Mr. Rajajee to come present his data, and I say that he has to defend the people, and they will not come to the rescue.
Respected President of Studies, examiners, and fellow research scholars. Today I would like to present my PhD thesis entitled Certain Studies with Antimicrobial Substances of Insectology, the relevant bioinformatics. The fundamental feature of the immunity is to distinguish self from non-self. Whereas the invertebrates rely on innate immunity because they don't have adaptive immunity like the vertebrates. Even though the invertebrates are successful from invading parasites and microbes, insects protect microbial invasion through cellular and humoral immunity. The key components of the insect immunity, in which the first defense mechanism is being shown by the critical. The first defense mechanism is being shown by the critical of the exoskeleton, chitinous and the Golgi factor. And as well as here the two components are being placed a major role in immunity, cellular and humoral immunity. Under cellular immunity, various types of the hemocytes are being playing the role of phagocytosis, modulation, and encapsulation. Whereas during the humoral immunity, phenol oxidase, lysozyme, transferrin, reactoxin, and antimicrobial are going to play the major role. So far, more than 250 antimicrobial peptides have been identified in insects, such as rifampicin, atacin, cecropin, ceftriaxone, and gamacin. The primary objective of my thesis is how does the innate immune system defend bacterial challenge in archaea to understand the role of phenol oxidase, lysozyme, and antimicrobial peptides in their relative gene transfer, in which the first we did is phenol oxidase, lysozyme role and their activity, and transcription analysis of three different genes related to the antimicrobial peptides and proteins. One is cobatoxin, and the second one is gallerinosine, and the third one is transfer. And we also evaluated low molecular weight peptides which has been expressed during the microbial infection in the human through gel electrophoresis, and their molecular weight has been characterized with the model of and relative bioinformatics studies. This is a test insect what we have used for the experimental purpose in my PhD. It's a lepidopteran worm and pest to the it's a major pest to the castor and is also pest for the various other crops. The experimental design which has been adopted for the work is we have reared the insect in our laboratory at 27 1 degree centigrade with a relative humidity of 65 with the 12 hours of day, 12 hours of light and 12 hours of the dark, and the microorganisms which has been procured from the MTCC, and which the gram-positive bacteria, Nagaraja and the gram-negative bacteria, E. coli, which has been procured from the MTCC Chandigarh, and they have been maintained according to the data sheet what they have been provided in the LV broth and LV agar. We have challenged the bacteria by suspending the bacteria in a prospect of a saline, and after challenging the insect by injecting the bacteria at different time intervals, the human samples has been collected 1 to 5 followed 24 hours. We have collected the human samples, and that human sample has been used for the antibacterial, antifungal activity, and as well as protein estimation. And as well as during, along with the human, we have collected the fat body to identify various kinds of the genes which are being involved in the transcription. Because fat body is the source for the hemolymph proteins. So we have also collected the fat body from the RNA isolation and cDNA synthesis, in which by using semi-quantitative RT-PCR we have analyzed the difference in ototoxin, galeromycin and transferrin. And we have also estimated the phenolphthalein activity, lysozyme activity, and the protein has been resolved to the crystallizing gellar proposals for the low molecular weight proteins, and each antibiotic activity modeled off has been characterized for the molecular weight determination. This is a detailed protocol how the insect has been cultured and the bacterial culturing methods by using the urea that we brought at 37 degrees centigrade for the E. coli and 30 degrees centigrade for the Macrococcus luteus. And all the experimental larvae has been grouped into four. One is the first one is the control larvae, which has not been challenged either with the bacteria or with any kind of a thing. And the second one is sterile injury to have a positive control because we are going to inject the E. coli and as well as the Macrococcus luteus suspended in the phosphate buffer saline by using a micro injector. So there may be intricacy of any injured proteins or the injured stimulations or the stress responsive proteins which may have a role in immunity. To have we have also have a second the positive one was sterile injured larvae, and the third group is of E. coli challenge larvae, and the fourth group is of Macrococcus luteus challenge larvae. After injecting the bacteria at different time intervals 1, 2, 4, 8, 12, and 24 intervals, we have collected the hemolymph samples, and it has been so by using a sterile anticoagulant having a composition of 90 mg of anti-OH and NaCl, EDTA, and as well as citric acid along with the protein which is of a protease inhibitor to prevent the coagulation, and as well as phenolphthalein to prevent the mineralization. At each time point 10 larvae has been used. And after collecting the hemolymph has been centrifuged to remove the hemocytes, and the pure hemolymph has been transferred into the sterile polypropylene tubes and has been stored at minus 20 degrees centigrade for the further analysis. And at the same time, after collecting the hemolymph from the same insects, the fat body has been dissected out in the sterile conditions by and has been transferred to a sterile polypropylene tubes treated with the DETC, and they have been stored and exposed to the liquid nitrogen and flash frozen and stored at minus 80 degrees centigrade for the RNA isolation. The total protein has been estimated by using the platform method by using Bowell cell amalgam as a standard. And antibacterial activity has been done with the radiodiffusion assay on Rujia bacteria, and antifungal activities has been done with the Potash using as well as the Snyder. And lysozyme activity has been done according to the Jaros using egg white lysozyme as a standard. And phenol oxidase activity has been done according to the stress team using 3,4-hydroxyphenylalanine as a standard. And the total RNA has been isolated from the frozen fat body, and the total RNA has been used for the synthesis of the cDNA. And we have obtained various immune-induced homotoxin. We have obtained the homotoxin, galeromycin, and transferrin gene sequences from the NCBI. And from those gene sequences, we have submitted for the priming for the designing of the primers to have an amplification purpose. And those primers have been synthesized from the MWG family. And these are the PCR conditions which has been used for the amplification of the cDNA. And the products has been quantified by using the, and as well as the densitometric analysis has been performed by using the image software. The gellar hypothesis, according to Shagar and Jabo, to visualize the low molecular peptides, crystallizing SDS gellar hypothesis has been adapted.